So today we have three speakers who will be discussing our latest product launches. Greg Porcheski, Priya Ramamurthy, and Andrew Swell. All three have been at Metabolon for some time and are experienced metabolomic practitioners. The agenda for today, I will start off by giving a quick background on Metabolon's product offerings and our technology. Then Greg will speak about the biological stress panel, Priya will introduce the liver panel, and Andrew will discuss the kidney function discovery panel, and lastly conclude with a live demo of My Metabolon portal. So our three most recent product launches complement our existing portfolio, from untargeted metabolomics to targeted metabolomics that include GCP validated panels. Overall, our aim is to build products that can answer a range of questions for academia, industry, population health, and applied markets. Where there can be profiling for differences between disease or high-risk patients with healthy controls that can enable researchers to underpin molecular mechanisms that govern a particular disease, or develop early diagnostic markers to complement clinical scoring systems. Or for commercial needs, Metabolon products can help develop novel metabolite biomarkers or drug target engagement, patient stratification, and disease progression. We have the capability to service a range of needs, so please do get in touch if you are thinking about using metabolomics. Our global panel and our discovery panels are built upon the largest level 1 library in the industry. For those unfamiliar with what that means, one of the bottlenecks of untargeted metabolomics is the annotation of metabolites. There is a wealth of software tools that can help you match your features with databases such as HMDB, GNPS, lipid maps. However, they will only be at best to deliver level 2 annotations. For a level 1 annotation, you must match your master charge with two orthogonal properties like retention time and fragmentation. You must match this with an authentic standard that has been run with the same chromatography and using the same instrument parameters to get the highest level of annotation. The immediate advantage of having level 1 annotations is consistent and reproducible results, facilitating quicker, actionable insights and saving you the time-consuming process of annotation. So with that, I'll pass on to Greg.
Thank you, Brennan. And as Brennan highlighted, we utilize the highest quality standards when it comes to generating data. And that is also true for the biological side. Employing a team of PhD biochemists with decades of cumulative experience, we identified the most pertinent metabolomic markers of biological stress by combing through hundreds of papers, utilizing our in-house institutional knowledge, and cross-referencing those findings against our historical detection rates so that we can deliver the nuggets of gold within your data set. On the next slide, we'll go over what we mean by biological stress. When I say biological stress, everyone in this room is probably thinking of slightly different aspects of stress. We can sum that down to three major aspects, ranging from stimulus, response, and consequence. The stimulus can include exogenous factors such as an infection, endogenous insults such as reactive oxygen species, or a mixture of those, such as in the case of a physical injury. A cell recognizes these stimuli either internally or externally, resulting in a response. Recognition of an antigen may result in the release of immunomodulators such as oxylipins, or changes in reactive oxygen species production may result in the expansion of the glutathione pool. This response then can result in compensatory changes or consequences, such as methionine being converted to cysteine to fuel glutathione production, or large-scale energetic shifts to fuel immune cell expansion. Coupled to response, we have resolution. The examples given previously are constantly happening in all of us, and resolution of these stimuli is the difference between health and morbidity, such as comparing a sneeze to an allergic response. Balance between these processes is what drives disease, intervention, and outcome. Metabolomics and these metabolites selected for the biological stress panel provide that deep level insight to examine stimulus response in consequence and resolution, which then can be used for drug screening, disease progression, identification of mode of action, or disease etiology. On the next slide, we'll get into a few more details. For instance, what we have here, you've probably heard of taking fish oil or krill oil supplementation. Why? For polyunsaturated fatty acids. And while we attribute most of the action to PUFAs, it's actually their derivative, the oxylipins, that confer most of the biological action. These are present on the panel. Whether you're looking at changes within the extracellular matrix in response to cancer within a tissue or within the skin, we can look at changes in collagen and look at markers such as hydroxyproline. When I said stress, a large amount of you probably had an increased amount of cortisol. So we can look at the amount of stress that's being experienced by an organism and how that may contribute to the overall etiology of the disease. As mentioned, glutathione. And these pathways can be divided into 7 discrete segments. And with that, I'll hand over to Priya, who'll talk about what we offer in the liver health space.
I think Priya's having some issues connecting, so maybe we'll go to the kidney panel for Andrew, if you're ready.
All right, so yeah, my name's Andrew. I'm going to talk to you guys about our kidney function discovery panel. So numerous metabolomic studies of kidney disease patients have identified metabolites perturbed with declining kidney function or kidney disease. And those include notable alterations in carbohydrate metabolism, amino acid, nucleotide, and lipid metabolism. For example, 2 common biomarkers of kidney disease are metabolites, urea and creatinine. So Metabolon's kidney function panel really expands on this. So we analyze 84 metabolites commonly associated with kidney function or disease, and very similar to the oxidative stress panel. We chose these metabolites based on our internal knowledge, as well as what was published in the literature, and then as well as detection rates. And some notable applications for this kidney panel include nutrition research, so whether a various diet or supplement affects the kidney, cardiovascular disease, diabetes, which notably has emerged as the most important risk factor for chronic kidney disease. Various aspects of cancer research are interested in side effects on the kidney function, and then also just in general, toxic side effects of drugs could also be a notable application. For this panel, we're looking at mammalian fluid matrices, so 200 microliters are required for plasma, serum, or urine. Next slide, please. So why do we care about function, or why is it important? Obviously, it's very prevalent. There's 1.7 million deaths annually, and that's worldwide. So 800 million people are living with chronic kidney disease. And again, that's worldwide numbers. Strikingly, 90% of people with chronic kidney disease are unaware they have it. And that's just within the US. There's estimated about 15% of people in the US that are estimated to have chronic kidney disease. So that roughly equates to like 37 million people. And 2 to 3% of most high-income countries' budget are spent on chronic kidney disease research or treatments. What's also notable is, as of February 2023, around 88,000 people are still on the waiting list to get a kidney transplant. So it's still a huge issue, and hopefully this kidney function panel can help address identifying early demise of kidney function. So a little bit more about kidney disease. Chronic kidney disease, as is often referred to, as a gradual loss of renal function that eventually results in end-stage renal disease. And this is important because by the time kidney disease usually gets diagnosed, it's generally too late to improve kidney function or restore kidney function. It has a high growing prevalence, and it's an important cause of morbidity and mortality among non-communicable diseases. So the big challenge for the kidney disease field right now is early detection. As I alluded to before, a lot of the biomarkers of kidney disease are late stage or end stage markers. So even by the time you diagnose disease, it's too late to treat it. And so what this panel will, provide is additional biomarkers that researchers can use to identify hopefully kidney disease at its earliest stages and can enable more timely intervention. It can also be used to gauge treatment response or understand side effects of a certain treatment. This panel could also help understand the underlying mechanisms of disease progression, hopefully uncovering early markers of early progression at a stage where you can potentially intervene and treat. And the current tests that are currently out there really fail to capture the complex metabolic changes that occur in kidney disease. For example, urea and creatinine, the two commonly most biomarkers, have numerous limitations. They're affected by hydration or supplement use or food intake. So there really is a strong need for novel biomarkers that are more specific, especially for at the earlier stages of disease. Next slide, please. So a little bit more about how this panel is structured. Again, it's 84 metabolites covering a wide range of nodes of metabolism. That includes amino acids and derivatives, various nucleotides, lipids, several metabolites associated with microbial metabolism, carbohydrates, several markers of protein catabolism or decay, and then lastly, several metabolites associated with nicotinamide metabolism. And so importantly, what these pathways can allude to is the biochemical pathways that point to the molecular or genetic drivers of kidney disease. In conclusion, the key benefits of this panel really include identifying the most appropriate markers of kidney function or disease. It can help address critical questions regarding mechanistic insights. It can aid in novel drug discovery. It can be used to predict adverse drug side effects. And lastly, many of these biochemicals can be used to improve the estimation of glomerular filtration rate, which is the measure of how well your kidney is functioning. And currently, a lot of the metabolites on this panel are being actively used to improve GFR. And with that, I will pass it back to Priya.
Thanks, Andrew. Today, I'm going to talk about the liver fibrosis panel. This panel consists of expertly curated 105 metabolites associated with the initiation and progression of liver fibrosis. This panel can provide or address key factors in liver fibrosis, including mechanistic insight, like early progression markers of fibrosis, also treatment targets, and drug discovery and testing. So listed here are the sample types that this panel is designed for, which includes serum and plasma and liver tissues. And we have also listed the areas of research this panel can be used for, but they are not just limited to these areas of research. Next slide, please. In the next slide, I'll go over why we decided to do a liver-specific panel. So the prevalence of liver diseases are increasing in populations around the world, and it has been accounted for 2 million deaths annually worldwide. In men, 66% of liver-related death is due to liver diseases. On top of that, due to the prevalence of chronic diseases like diabetes and obesity, 25% of the world's population now has non-alcoholic fatty liver disease, which, if left untreated, can lead to advanced liver diseases like cirrhosis and hepatocellular carcinoma. On top of that, the economic burden these conditions pose on our health system is also mounting. We spend around $135 billion annually to manage these conditions. So there is a clear need for early markers of liver fibrosis to address these issues related to liver diseases. So let me move on to what is liver fibrosis. Liver fibrosis is a condition where excess scar tissue accumulates in the liver due to chronic inflammation, which then leads to serious liver damage and diseases. And this causes morbidity and mortality worldwide. So liver fibrosis is again a progressive disease. If left untreated, can lead to advanced liver diseases like cirrhosis and hepatocellular carcinoma. So early detection is paramount here to prevent progression to advanced liver diseases and also for effectiveness of treatment. This early markers can also help us understand the mechanism of liver disease progression and potentially uncover a drug target for treatment. So, what are the limitations that we are facing with the current markers and tests that are available for liver fibrosis? For example, if you take liver biopsies or imaging, they are very expensive in nature, and there is also lack of early stage detection of fibrosis. On top of that, these are very subjective in nature. Another standard test is enzyme assays like ALT and AST. Again, these tests lack early stage detection. Since liver pathogenesis is closely associated with metabolic dysfunction, metabolites can be leveraged as biomarkers for liver fibrosis and understand liver dysfunction and move on to find mechanisms of early disease progression and target identification. Next slide, please. So with our liver fibrosis panel, we have expertly curated molecules from literature which are closely associated with liver fibrosis. These 105 metabolites are then organized into 7 different pathways, which can easily provide the stage of fibrosis progression. For example, if you take inflammation and oxidative stress, these are the causes of liver fibrosis, and this will kind of allude to early stage of liver fibrosis, while your ECM remodeling is the scar accumulation phase where fibrosis actively takes place. While the next 4 pathways are associated with the effect of liver fibrosis, which include ceramide, energetic, bile acid, and lipid metabolism. So using these 105 metabolites spanning across these different stages of fibrosis, you can get insight into whether it's early stage fibrosis or late stage fibrosis. With that, I will wrap up with key features and benefits of this panel. One of the key features of this panel is the coverage of different pathways which are associated with the stages of liver fibrosis. And the processing of this panel is also quicker, and it also has an option of a non-invasive option when you choose plasma or serum. So to end, this panel will provide researchers more confidence in identifying appropriate biomarkers for liver fibrosis, which can help address critical questions associated with the mechanism of liver fibrosis progression, target discovery for drug treatment, and testing. With that, I'll pass it to Greg to go over the deliverables and visualization for these products.
What presentation isn't complete with a live demo? And we've told you all about the great things that we do in terms of data quality, But let's get into what this data actually looks like. What are you going to receive and how are you going to utilize this data? After you would log into our portal to access your data, you'd be met with this screen, which would start immediately displaying the results of your study of interest. What I have here is some samples that Metabolon purchased that were related to COVID and inflammatory status, and we're looking at the results of that study. I'm going to anchor us to these tabs at the top here. So on first, we're going to look at this Discovery Panel report, which is going to give us a broad overview of your results. And then we'll move through each subsequent one to see what kind of analysis we're providing to you with this product. Right below it, we're going to select our comparison of interest. So if you have, for instance, a control group and a treated group, you'd have a single comparison. If you were to run a two-way ANOVA with two factors, you would end up with 4 pairwise comparisons, and so on and so forth. So once you've selected your comparison of interest, you could evaluate how strong and feature-rich your data set is. If you have a relatively weak data set, you could adjust your p-value threshold, for instance, and you would see those changes within the display changing in real time. By default, we go with that 0.5 p-value threshold. The results are displayed in several areas. On first, what we have here in the center is what we call our bubble plot, where you can kind of see where those changes are grouped together based off of how we've classified these compounds. So we have here as an explanation where the size of the circle reflects the p-value, with a smaller p-value reflected as a larger circle, and then the directionality of fold change reflected by the color. So small fold change due to the light kind of blue color, but very large circle, very strong p-value. And inverse here where we have with the red with the, going in the other direction where it's more abundant in our numerator. We can already see here that between these two groups, we have different processes that are being activated, from amino acid immune modulators to oxylipins, differing between these two groups. So already we're kind of getting an idea of what's different between COVID and other inflammatory disease states. There were a lot of news articles and publications on, for instance, the interplay between the bradykinin system and COVID, and we already are seeing that observation right away. On the left side here, we can see the detailed information about what's going on with individual metabolites highlighted, their p-value and fold change for this specific comparison. We can also get that information by highlighting these individual elements in the center here. These are also sortable. So you can see if you want to look at the largest changes, the most statistically significant, or if you're just looking for a particular one going by alphabetical, or searching for a given compound, have it highlighted so you can find out where this is on this map. In addition, we have pathway enrichments that are provided here. So while an individual metabolite may belong to a single pathway in terms of how we've grouped it above, a single metabolite may actually belong to multiple biological responses involved in aging, coagulation, mitochondrial function, and so on. We can see how many metabolites are in a given pathway, how many of them are significant, and that percent significant. So very quickly, we can see, okay, smooth muscle function, Warburg effect, and vascular function, hypoxia, and anexia are being impacted within these comparisons. I'm already making some connections between the vasculature, which delivers oxygen, and the connection to hypoxia. I did not actually prepare for that section of this, that I just did right now. I'm making findings live in front of you. Next up, we're going to go to this Impact Explorer. Most studies have multiple comparisons that you want to examine side by side, and that can become problematic for something like a bubble plot. What we've done is we've taken a typical heat map view, circularized it, and overlaid another piece of data. Let me explain. So what we have here on the outer ring is an individual comparison. Each of these dots along the center represent a specific metabolite. So this box here represents adenosine and the statistical results within this comparison. These boxes on the inside represent these biological processes. So for instance, on a panel that looks at biological stress, we have a lot of overlap with inflammation. A lot of these metabolites obviously are related to inflammation, but we can see exactly which metabolites we think are associated with inflammation and have those highlighted for you. We'll also highlight which one of these are statistically significant across these comparisons of interest and in which direction they're going. We can also flip what we're looking at, and if there's a specific compound, so for instance, I see the sphingosine is having large changes between these groups, I can see what we've annotated that to be related to. Neurological function and apoptosis, for instance, here. And we can get an idea of how strong that neurological function signal is and apoptosis. So with this, we can look at multiple comparisons, have this information updated for us in real time, see what those results are, and have those connected to biological processes. Next up, we have the profile finder. If you've identified an area of interest through either of any of these maps, you can now explore those a little bit deeper and looking at the biology by going into the profile finder. What we have here are high detailed figures that go over the connection between a specific metabolite, how that message is relayed, and the consequences of those metabolites. In addition, we'll provide a write-up that kind of connects those findings in this area to biological stress. So you're not just left with, okay, Metabolon said this is related to stress. What do I do from here? You'll have those resources at your fingertips so that you can take that farther and so that you can explain it to the next person, whether that be in your grant submission or your publication. Last up, we have the ability to download the data. What I kind of showed was analysis in terms of what additional things we can provide here. But the first question we often hear is, I want the raw data. I want the area counts. I want to see this. I want it in Excel. I'm used to Excel. I like Excel. And I'll show those in a moment once we're done with what's on the website here. Last up, we have a support section, which goes into details about everything that you can be looking at. So you can have an understanding, or you may have frequently asked questions that you can get addressed. I know I just gave a brief explanation of, for instance, the Impact Explorer and the Heliogram. If you were to use this product, you may have more questions down the line. We have videos that explain this content. We have material that in text will walk you through this so you can understand what you're looking at without having to wait for someone to respond, though we're there to help as well. I'm going to switch now to those files that I mentioned that you can download. What I have here is what we call our data table. I typically refer to this as our machine readable file. And you'll very quickly see why I call it that. It plays very nicely with most omic pieces of software. We have an explanation tab to help anchor you and ground you should you forget what you're looking at. After that, we have all of our chemical annotations. So everything that we've detected, how we've grouped it, external versus internal identifications such as SMILES, to KEGG links, to HMDB links, so that you can see what other people out there are saying about these metabolites. Your sample metadata as collected from you and provided to us, as well as what we've collected on your samples and providing back to you. From there, we start getting into the actually collected data. At the top here, what we have is an individual metabolite identified by its chemical ID. And on each row, what we have is the individual sample. So this sample and all of its detected metabolites can be seen here. From there, we have various statistical operations that are performed on the data set for various reasons, which are explained within the explanation tab. And I'm going to switch now to what I call the human readable file. Again, we're going to start off with, there is always going to be different ways that we communicate explanations to you. You're never going to be wondering, what am I looking at or how to use it? We'll have things written and it'll always be an email contact away. But what we have here is the results of that analysis. So for instance, the same comparison that we saw on the portal is shown here. We have that same kind of information as it relates to, for instance, HMDB links and KEGG links, as well as the individual metabolites and the results of that comparison. I'm going to zoom in so this can be seen easier. So for instance, if I was curious about an individual metabolite and I wanted to work at this in Excel, I could see the fold change between the various groups. I could also look at my p-values and q-values, which are how we correct for multiple tests being performed, to the mean values and percent fill values. Included in this are various summaries of the data set, including a statistical comparison that looks at the total number of differences. It's very similar to our bubble plot, and even quality standards such as RSD of internal and endogenous metabolites within your set. And with that, that completes the live demo that we have right now.
Thank you. So we have a few questions, so I'll try my best to get through them. So first question is, what are considerations choosing between serum and plasma samples for human studies? Greg, do you want to take that one?
Sure. There are a number of considerations when choosing a matrix, and that question probably arose because on our various slides, we include plasma and serum. So do we have a preference? Should you have a preference? And so on. The first question I always ask is, be future thinking. If you're doing a study today, what does tomorrow look like? If this is a success and you're going to plan to use a biobank that has a number of samples in your area, identify what samples they have. Try to make your samples like those samples. If they're using serum in the biobank you're interested in, send us serum samples. If they have plasma, do plasma. From our end, they're very similar, and we can supply a white paper to that effect. But if you have no preference, plasma is usually our go-to for what we recommend.
Great. And then another question would be, is it possible to run multiple matrices together? Would urine and plasma data be comparable if run together in a project? Since it's got urine included, Andrew, would you
I can take this one. So for the discovery panels, we would like to only analyze one matrix per project. So you can analyze your set of serum samples as an independent project, and you can analyze urine as an independent project as well. We can't analyze them both together at the same time under the same project. So we can analyze the different matrices, but they have to be independent projects or sample sets.
Great. Great. Okay, next one. Are metabolites in these special panels not covered in the global untargeted panel?
I can take that. The metabolites in this panel are covered in the global panel as well. All the metabolites in this panel are under the global panel as well.
Yep, I would just add that these panels have been based on Metabolon's experience over the last 15 years, relating these metabolites to particular diseases, along with the literature as well. Okay, how do I access your platform for my metabolome analysis? I want something different from MetaboAnalyst. Greg, you want to take that one?
When there's a couple of questions there. So one is, if you have your own data set, are you able to incorporate that into our workflow? Unfortunately, the answer to that is no. If you were to do a project with us, how easy is it to load into our system? There is no loading into the system. It's just inherently done for you. The second you come on, you will see your results within that pathway. So we've tried to streamline that as much as possible. That live demo I was showing, that was what you'll see the second you press login and you click your project. I only didn't show me logging in to protect my password and such, but that is the first screen that you would see when you look at your study results.
Nice, great. Does Metabolon provide interpretation of results for all panels? So the biological stress panel, the kidney panel, and the liver panel will have access to my Metabolon. Can your platform be used only for human metabolomics? What about bacteria and fungi? Can it be done individually?
I can answer that. Yes, we do metabolomics on bacterial pellets and fungal pellets as well, and they can be done individually as well.
Okay. Next question is, do you have a panel for heart tissue for cardiovascular research to look at cardioprotection? What would you recommend?
I can take that one. Yeah, unfortunately, we don't have a cardiovascular specific panel at this point. We do have a lot of metabolites that we know are related or highly associated with cardiometabolic diseases. So at this point, I would recommend going with our global discovery panel, and then a study director like myself can interpret the results with a particular focus on identifying metabolites related to cardiovascular disease or cardioprotection.
Yeah.
I'll add to that. We have experience with a lot of cardiovascular tissue projects.
Yeah. Okay. Do you all have any advices into annotation of unknown metabolites from untargeted metabolomic study? Any tips into manual or high throughput annotation using free databases? Well, my first advice would be to be very patient. It is a long process, and it can also be different between different users as well. So using HNDB, lipid maps, GNPS are all free resources. Metlin is now a paid resource. But I still think the easiest way to just get immediate results would be using a provider. And I will also add that when looking at a provider, other providers, it's important to realize that they can advertise a high number of metabolites. But what this really means is just the number of features that are detected in your instrument from your instrument. You have to go into annotation, and Metabolon has gone the extra step further to match features with authentic standards so you can get level 1 annotations. So what we advertise is the number of metabolites in our library, not the number of features that can be detected. That the number of features that can be detected will not give you biological interpretation. You need annotation. Does Metabolon support any flux analysis labeled metabolomic studies, 13C or 15N, or combination of both?
I can take that.
Okay.
Right now, we do not have flux analysis. So we do global metabolomics and these targeted panels. And we do have targeted panels for specific metabolites, but unfortunately, we do not do flux analysis right now.
Okay. Is there any difference in accuracy of the data for plasma metabolites if the blood samples collected in EDTA or ACD collecting tubes? And or does it not matter with the collection tubes? Different.
I can take that. For the most part, the profile is very similar with minor differences based on the coagulant used for it. So, I mean, it's less than 10%. So I would say it depends on what you availability of the type of sample you have, whether it's collected in a site where they only collect EDTA plasma or a site where they only collect serum. So the differences are less between EDTA and plasma and serum.
Okay.
Yeah, and I'll just and I'll also add like some of the metabolomic differences between plasma and serum. While relatively subtle, they are important to consider. So with serum, you're generally going to have higher levels of polypeptides, peptides, and other metabolites that are likely contributed from the clotting. So you'll have higher content of just various peptides and protein fragments. And then in general, serum has higher levels of metabolites as compared to plasma, yet they all still are highly correlative. But serum, the higher levels in serum can make it more sensitive for biomarker detection. And then, of course, serum doesn't have fibrinogens or the clotting factors where plasma does. So those are some, and then plasma generally has lower levels of inflammatory mediators as compared to serum. So those are some important things to consider when you're choosing serum versus plasma. But between the different types of plasma, between EDTA or citrate, they're very similar. And there's several publications within the last year or two that are comparing serum versus plasma that we can hopefully share.
Great. Thank you. Okay, next one. Do you find that a targeted panel, for example, liver fibrosis, fatty acids, etc., would give a more specific analysis in those components compared to the untargeted discovery panel?
I can take that.
Yep.
In untargeted, it's global. So we are focusing on the research question you're asking, while your liver panel or fatty acid kidney panel, it's focusing on markers specific to that area of interest. And these are all like well-curated metabolites associated with these, panels of interest. So I think you will have a specific focus with these panel compared to the global, where you will need a study director like me go picking on signatures that are associated with the research question you're asking.
Great, thank you. If we use your platform, should we follow certain standard procedures for the blood collection/storage at the beginning, including tubes?
Yeah.
I can take that. Oh, go ahead.
The most important factor here is consistency between your samples. If you're going to be doing it one way, make sure you do it that way for all your samples. Having said that, we do want to have them frozen as quickly as possible. So after, if you're separating for serum or plasma, once that separation is done, that you freeze that in -80, and we can also provide you tubes for shipping to us to automate that procedure.
Great. And do you do metabolism on, or probably means metabolomics, but do you do metabolism or metabolomics on human cells like human corneal epithelium cell line?
I can take that. Yes, we have done metabolomics on human corneal epithelial cells and lens as well. So yeah, we have experience with the cell types as well.
Great. What are your recommendations for sample collection, transport, processing, and storage? I think, Greg, you just touched on that on the previous 2 questions before.
Yep. The important thing is to be consistent. And then when you work with Metabolon, we'll send you an actual sample collection guideline document that you can follow. And it'll list, our recommendations.
Great. Okay. Do you have any experience to analyze bacterial culture media? Is that possible to find pathogenic metabolites?
Absolutely. So we have quite an extensive amount of experience, not just in single culture system, but co-culture or multi-culture systems, where we have multiple organisms that can be interplaying together to result in a pathogenic response. This includes popular things like C. albicans, as well as more exotic growth pathogens. With that in mind, looking at the serum, the media in this case is pretty straightforward to do. And yes, we can correlate that with pathogenicity.
Okay. Okay. Any advice on whole heart collection for metabolomic study?
Yeah, I can take that one. So we've done several heart tissue studies. In terms of the whole heart, if the general recommendation is to try to keep them roughly the same size, or if you're going to take a chunk of it, keep it roughly the same size. But in general, just flash freeze the heart as quickly as possible with no additional additives. And then, just be consistent and keep that heart, unfrozen until metabolomics.
To add to that, our recommendation for storage is -80. Store it at -80 till you ship it to us on dry ice.
Yeah, and depending on the size of the heart, we usually only need 50 milligrams to 100 milligrams of tissue. So it depends on your type of research, whether you need to really send the whole heart or if you can just send a portion of the heart. But as study directors like, my Priya and Greg and I can help you assist with a lot of these questions up front before you do proceed with a study with Metabolon.
Okay. Do you have specific panels for cardiac energy metabolism? A lot of interest in the heart, cardiac.
Yeah, definitely. So fortunately, I've actually interpreted a few cardiac studies recently, both cardiac tissue and the plasma serum from, subjects or animal models with cardiac disease. So I would say the big ones are acylcarnitines, which are the markers of fatty acid oxidation. Free fatty acids are another big marker. And then I would say creatine, creatinine, creatine phosphate are especially important to cardiac tissue, as those metabolites are kind of the key energetic reserves for cardiac tissue. So when you assess the levels of those metabolites in the tissue or the plasma serum, you can get a sense of how your cardiac energetics are shifting. And then additionally, free amino acids. You've got a lot of input from amino acids into the heart for energetics.
So, like adding to that, the global panel would be the appropriate panel for cardiac function right now, because we don't have a specific panel for cardiovascular diseases.
Yeah, and the global panel, the nice thing about the global is going to cover all those aspects. So it's going to give you how is glycolysis changing in the heart, is fatty acid oxidation going up or down in the heart, are amino acids contributing more to the TCA cycle. So these are all things that the global panel can uncover, and then a study director like ourselves can help you interpret and give you additional insight.
Okay, next question. Does Metabolon plan to provide other services for spatial metabolomics and or single cell level metabolomics profiling? So right now we're provide global profiling, so untargeted metabolomics and targeted panels at the moment. But we do welcome all inquiries, and it'll be useful to understand your study objectives as we may be able to help you achieve your goals with our current services. Okay, next question. Do you have any panel or workflows to detect food metabolites in serum or tissues like colon or liver?
We can definitely work with tissue samples, colon or liver. There are a number of metabolites that are specific for dietary changes, but there is no way to distinguish necessarily between a metabolite that's shared. Dietary glucose is no difference between circulating glucose, but phytoestrogens are not synthesized by humans and so are ingested. There are a number of compounds like that that must be ingested that are part of every single cell process, branched chain amino acids, for instance. So there are ways that we can look at the data set, depending on what we're looking at, whether it's the liver or even fecal tissue, that we can identify what's going on with the diet, identify dietary differences from uptake to even composition.
Great. Okay, about the heart again. It's all good asking the questions. Rat heart weight is about 1 to 2 grams. Is there a way to send you all the heart without dividing it into parts?
Yeah, so I mean, we can analyze the heart. I mean, our sample prep team would likely have to process these samples into smaller amounts. So it's, if you're comfortable with our sample prep team dividing the heart into equal amounts, then we can process these. My recommendation would be for you to divide these beforehand so you can save the extra portions of the heart for other types of research. Or we can also divide it and then ship you back the remaining tissue amount. But that induces a lot more freeze thaws, which we generally do not recommend.
Okay, I wonder your opinion about single cell metabolomics or metabolomics with low quantity cell number. Any idea for the minimal number of cell that can be detected? Its intracellular metabolites. So I've previously done cell culture work and I've gone as low as 500,000, but I would definitely say to be safe and to be reproducible, you should go 10 to the power of 6, so 1 million cells.
Also depends on your assay type and what you're looking for and what you define success by. As you reduce the amount of material submitted, you're going to get less metabolites detected. Some studies, they're okay with having 200 to 300 metabolites detected. There's really key things that they're looking for, and they're the most abundant markers. If you're looking for those subtle changes in low abundant compounds, we're going to need to see more material so we can detect that. Targeted assays versus global also have differences in sensitivity. So depending on the approach that you take, you may be okay with getting lower and lower amounts. Brennan's suggestion, though, of about 500,000 cells is where we'd like to see really the minimums.
Okay. Does the global panel cover environmental chemicals?
Yes, I think some of them, like PFAS, tobacco-related metabolites, but it depends on the level of exposure as well. So we do have some of them in our library, but the detection will depend on exposure and accumulation of these in the subjects or in the mice.
Okay. So someone said, basically, if I have already an idea of the class of metabolites that should modulate my phenotype, would you suggest a targeted panel compared to the discovery or untargeted panel? So I would say it would depend what those class of metabolites are and what your scientific question is. Do you want to quantify your results or do you want to get a semi-quantitative result as well? Metabolome can cater for both, but yeah.
Yeah, that really just depends on your phenotype that you have at the moment. If, for example, you have a phenotype that's really a fatty acid oxidation signature, and that's your sentinel signature that you care about, then a targeted assay at Metabolon for just the markers of fatty oxidation is probably the best approach. But if you have kind of an overall, if you're still kind of in hypothesis generation mode and you think you have a fatty acid oxidation signature, but you want to see if there's other nodes of metabolism change, then our global discovery is the better approach. But like Brendan mentioned, if you want absolute quantitation, then a targeted assay is the best approach, whereas the global discovery is the relative quantitative data. But yeah, and a study director like ourselves can help you guide you to what targeted assays we have. And feel free to check out our website as well, which has a list of our current targeted assays.
Okay. Another question about dietary metabolites. Do you have a panel to profile crop oligosaccharides?
Ooh, that's a tough question.
I don't think we have a specific panel, but I think oligosaccharides are very hard to capture because our molecular weight ranges between 100 to 1,000 or 1,200 Dalton. Some of the oligosaccharides are beyond that. So we don't have a specific panel, and we can profile some disaccharides, but not a lot of oligosaccharides.
Great. And I think that's the last of the questions that we have. But if you did want to submit questions or send in inquiries, please do that. And we can begin a discussion with DTS members like Greg, Priya, and Andrew, and they'll be able to help you out a lot more. But thank you to our speakers, and thank you for joining us today. And look on our website for the next webinar that we'll host. Thank you.